All right, welcome to this session. My title for the session was Fighting the Big C with the Big D, but Amazon marketers turned it into solving big problems with big data. But just to get an idea, this is in the AMPLab, which is a big data project at UC Berkeley. How many of you here saw the talk by Matej Zaharia and Ben Franklin? Mike Franklin? Yes, okay, so a few of you know about it. All right, great. So we started this lab about a year ago, and I'll tell you a little bit more about it. And it's about big data. We're going to build an open source data analysis stack and following the tradition of Berkeley Unix and Postgres and systems like that, make it available open source. But to create this technology, we needed to have big data. And for us in academia, you know, it's either really interesting and tiny or it's really big and dull. And if it's really big and interesting, it's probably proprietary. So we got started to try and find big data, and we hit upon this cancer genomics, which is what I'm going to tell you about. So I'll motivate this by what's the good news and bad news about cancer. There's actually good news as well as bad news. So, but the bad news is pretty bad. There's this Pulitzer Prize-winning book that came out a year ago called The Emperor of All Maladies, and it talks about the history of cancer and over the centuries what treatments have been done. But it's just an amazingly pervasive disease that the numbers are staggering. You know, it's the second leading cause of death in the United States. It will soon be the leading cause. It will take over from heart disease. 1/3 of all women in the United States will face cancer sometime in their lifetime. 1/2 of all men will face cancer sometime in their lifetime. There's 1.6 million new cases every year. There's 13 million just in the United States living with cancer. That's like 5% of the adult population. So the numbers are staggering. So that's one of the bad news. Now the good news is now, as this book records, we know that it's a genetic disease, that it's the changes to the genome that lead to these cancers here. And another great reason, coincidentally, thanks to the Human Genome Project, there's been huge investments in trying to bring down the cost of sequencing. Oops, I skipped. I skipped the part about that. What happens is when your cells replicate, accidentally there'll be some changes to your DNA, just random chance roll of the dice that can lead to the flaws that cause cancer. Or if you do carcinogens, so if you smoke, that changes your mutation rate a lot faster. You're much more likely to get cancer. But the good news, as I was saying, is the dropping in the cost of the sequencing. So this graph to the right, which is real data for the blue area, is over a 6-year period, the cost of genome sequencing dropped by a factor of 10,000. So Moore's law is like a factor of 10 every 5 years. This is way, way faster than Moore's law. So soon we will see this come down the projected line, get it below $1,000 a genome. And when that happens, all of us with health systems could afford to have our genome sequenced. And now, well, that sounds good. So who's working on this software, and who's working on collecting the data and that stuff? Well, that's some of the bad news. The people building the DNA software are scientists, and not computers, without the word computer. They're kind of biologists. So they don't have any, they don't believe in training in software engineering. And so the rate that this software improves and the quality of the software has some problems. Because it's not improving fast and it's not really fast, soon the cost of the data processing will be higher than the wet lab cost. And finally, no one's collecting all this data and putting it in a giant repository along with patient outcomes so we can mine it and get good results. So let's assume sometime in the future that these problems will get fixed, and we'll talk about when in the future. So this book, the author projects a better world for cancer patients. His theme is breast cancer. So he talks about the person who early in the book had breast cancer. In the future, goes to the oncologist, she has breast cancer, and she comes in with the cancer genome sequence with her. And they've organized all the variations, what's different about her genome and what are called pathways, which I'll explain a little bit more, but these chemical processes that lead to how your body works. And he runs it on this software, and it identifies the key pathways that are determining the cause of cancer in the future. And then therapies are targeted exactly to go after those pathways after that tumor is removed. as we'll say, cancer changes over time, and just like in AIDS, you take a cocktail, not just one drug to do it. She would be prescribed with a cocktail to take to get the rest of the cancer cells that the tumor, after left in the tumor. But these cells will change over time. She comes in, say, every 6 months, changes the cocktail, and may well have to take some kind of medicine the rest of her life. But like AIDS is today, the cancer would be controlled or possibly eradicated in this bright future. So that was great to read that part of the book. That's reassuring given the numbers. The not so great part about the book was when this was going to happen. So he said, well, imagine the future. And he said 2050. 2050, this thing will happen. So, well, that's great for those of us alive in 2050. That might not include me. What can we do? Can't we do something sooner than 2050? And that kind of led to this project. So here's the challenge, and I wrote this up in an article. It looks to me, and to some of us, that since the software is so important to a lot of humanity, we ought to get some people with professional training in building software to try and build this stuff. So let's see if computer scientists could get involved in building these genome analysis pipelines so that it'd be fast and accurate. And then, if that's possible that the computer scientists should get involved to do this, I kind of made the argument, well, maybe we could help. And then I thought about it overnight as you think about these numbers. Well, if there's a chance, if there's just a chance, maybe we can't help it. If there's a chance, aren't we kind of, as moral people, aren't we obligated to try? So that kind of got us hooked on it. I don't know if we'll be able to help, but boy, if we can help, it's so important. How can we not try? So the two things that, and the talk will be split up along these two pieces, two places you could help, we could help, would be to make one of these genome analysis pipelines so that most of the cost or the delays won't be in the software. It'll just be the wet lab pieces will bring the, I'll talk about the first part. And the second would create this giant repository that would have the genetic information and the patient records and outcomes and things like that. I'll talk about both those pieces, both of which computer-trained people could play a big role. Let's do Genetics 101. You get half of your genome from your mom, half of your genome, your DNA from your dad. We know about the double helix. It's double-stranded, and you can see the diagram on the right. There's bonds in between those two pieces, the little red pieces. What those things are called is base pairs, and the alphabet of the genome is 4 letters. For computers, it's 0 and 1, and for the DNA, it's G, C, A, T, and they're connected together at A's and T's and G's and C's on opposite ends of those pairs. And the human DNA has 3.2 billion of these base pairs. What's a gene then? It's called genetics. Well, this is this unit of heredity that maps onto these stretches of DNA. That we have about 25,000 genes, and on average, although it varies a lot, it's about 27,000 base pairs. And so in terms of keeping the cells alive and growing, the metabolic role of these genes, what do? They produce an enzyme. So for every gene produces one enzyme, and that helps control the biological processes. And this pathway word that was talked about is this series of chemical reactions that help maintain the organism. So we want to try and attack the pathways that are keeping cancer alive is what the hope is. And by knowing the genome of the cancer, that'll help us figure out how to attack the pathways as a result. The cancer is, normal cells, there's limits on how fast you can grow and how many times you can grow. The cancer is this bizarro cell that it has no limits on growth. It's immortal, it doesn't have a built-in death, and it mutates, it changes itself. That's why it's such a pervasive disease. And the cancer tumors themselves aren't one homogeneous set of cells. They're a colony of different types of cells. So classically, the doctor or the oncologist will go after the main part of the tumor and wipe that out, and they say they won't find any evidence of it, but there'll be some cells left behind that mutate and grow later, and that's why you hear those stories so often. And then it's got this terrible thing that it will metastasize. It will try and spread throughout the body and form other cancers, other parts of the body. And again, it's the pathways that you're actually attacking with the therapies. Okay. So let's talk about the sequencing of these things. So actually the way these machines work is they do chemical processes, and they end up with copies of the base pairs that are about 100 or 150 base pairs long. You have all these little pieces. and your job is to assemble them. Now, we have the reference genome thanks to the Human Genome Project. So what you can think about this is like doing a puzzle with a puzzle box here. So those are the pieces of the puzzle at the top of the slide, and the puzzle box you can look at to see what the puzzle is supposed to look like. There is the reference genome. So what we're trying to do then is, and then of course, that's the reference genome. We could just, every time we sequence, just give you back the reference genome. It'll be close. We're probably within 1%. All of us in the room are probably within 1% of the reference genome. So that's, we're 99% of it there. But obviously, the key is to figure out what's different about us. And those are called variants. And those little red dots represent the changes from the reference genome. And then we try and align them. And we do it not just one. It's actually lots of copies that line up with the genome. So we'll put all the ones that are supposed to align, and then we just vote, and we'll declare, well, the majority of the ones that align say there's a variant, and so we'll call that one a variant. And that's what this process is. This is called alignment to the reference genome. And when we're done, we report the variants. We can either report the whole 3.2 billion or just the changes from the reference genome, and that's what's unique about you. Okay. So classically, these genetic pipelines I was talking about have these 3 steps. You get the information from the machine, you try and align them, and then they do 2 classic steps. The first one is called SNP, for single nucleotide polymorphism, and that's basically, in our terms, like single bit errors. So one base pair is different from the human genome. And then, the second part is much harder, and it's called structural variant detection. When the DNA replicates, it's not just single bit errors. There's this process where it entwines and goes back together. So there can be sections missing, deletions, or sections inserted into the wrong place. Insertions, they can be reversed. They can be hundreds of base pairs long, thousands of base pairs long. So this is the way biologists have broken the thing. The SNP thing is kind of the easier step, and they've made good progress on this. The structural variant calling, to our surprise, is wide open. There's not a lot of understanding of that. So that was a nice, simple 3-stage pipeline that I showed you. It doesn't look too complicated. This is what reality is today. The software that's been developed by scientists to put that together into a workflow. This example has 119 steps, this person who reported this. And that workflow, that's these 119 steps that he has to accomplish, takes him about a week. So if he does everything right, he can get a sequence out, and that's what it's like today. And he gave this talk kind of showing how hard it is and how difficult for him as a researcher to get sequencing done with today's tools. So that's like, wow, that seems promising. So that's why, well, maybe we could help. Since I got involved in this about a year ago, I've learned about the kind of the quality of the software, and this apparently is well known. There was an article in Nature that talked about the poor quality of software developed by scientists. So in a recent survey, most scientists are self-trained, and only a third of them think formal training in software engineering is worth anything. I don't need that. 2/3 of them, I'm self-taught and I know what I'm doing. And then, pretty clearly from the survey, less than half of them know what testing is. So they're building the software, they think they know what they're doing, and they don't test it. So what's the consequence of this? So a professor at UC San Diego in the Scripps Institute, he got this software from another lab, published papers based on their software in very prestigious places. Science magazine is the cream of the crop if you're a scientist. All your friends shake your hand when they see you if you have an article in Science magazine. It's a 3% acceptance rate. Every 30 papers submitted, they accept 1. Well, he had to withdraw papers. He had to withdraw 5 papers, including ones in Science magazine. In my 35 years, I've never had to withdraw a paper. I never want to withdraw a paper. And imagine that you have to withdraw a paper, and it's not even kind of your fault. It's this other lab. So that's kind of the state of scientific software. This is a reason that people who get software training and believe in testing could play a role. This is what the team looks like. There's the UC part of the team of the faculty on the right, and the students and postdocs on the left. And we also have collaborations with Microsoft Research and with Intel, and they're in and UCSF, and they're in the middle. The color coding is the blue colors are computational biologists or people in the medical field. The machine learning people are in green, and the systems people are in gold. So about 60% of the people involved are in our systems. So we're kind of systems heavy. For those of you who missed the talk yesterday, a quick thing of what the AMPLab is. The AMP stands for Algorithms, Machines, and People, and what we mean by algorithms is machine learning algorithms, and we've got a very strong machine learning team involved. The machines is cloud computing, and that's the systems people. And the new thing is the P is the people is trying to do crowdsourcing. It started last year. It's actually going to go probably through 2017. We're going to do this open source stack. Amazon is a major founding sponsor along with Google and SAP, and we've got major funding from both the National Science Foundation and DARPA, and there's another 15 companies that are partners in it as well. So that's what we're doing. We're trying to do this stack. What the plan of record for the genome piece is, we will use AMP technology to be able to build our stack, and we believe by using much more productive tools, we'll be able to build a better quality software, a lot fewer lines of code, and be able to make it more maintainable. The system that was talked about yesterday is Spark. This is a kind of a library with an API that's been designed to go with Scala and Java, or there's going to be a Python version sooner. It relies on keeping things on memory, what they call resilient distributed data sets in DRAM rather than on disk. So they literally run 100 times faster than MapReduce and Hadoop. There's also Hive, which is the SQL interface that they've ported to Spark, and they're getting similar giant speedups as well. And so we're going to rely on Spark and Shark and the Mesos, which is a cluster operating system, and the rest of the BDT to develop those tools. So kind of why are we in the AMPLab? Because we're there to provide an application, and so we say what we need from these tools, and so that they can evolve in better ways when they make decisions. We're going to follow, because people want to add things to these pipelines. It's still early in the biological processes. We don't really know everything we need. It needs to be extensible. So like many people, including Amazon, we're going to follow a service-oriented architecture. So network interfaces with all the components rather than direct linking. And we believe in software engineering and believe in testing. We'll do continuous integration testing and have well-tested tools. The first example is going to be that first stage of the pipeline. We have something that's called SNAP. It's our nickname for Single Nucleotide Alignment Program. I'll show you about its speed, but it's a hash-based system. So you would get a read, and the piece of it, the first piece of it, so many bases that would index the design, and that would say that it's in a couple locations, and you would check those locations to see which is a better match. The read itself can have these bit errors in it, the SNPs in it, so we actually will go and make many samples to the hash table to do it. How does it compare with the standard aligners? They're a little, on the screen there's 4 very popular ones that are listed at the top in white and snaps at the bottom, and you can see it's about, in percentage aligned, it's about as accurate as the rest of them, and the error rates are very low, but it's tremendously faster. What can take some of these systems days to do the alignment, we can do in less than 2 hours. So that's our first result, which is encouraging about that by bringing our skill set, we can make a contribution. What we're doing next is try to improve that accuracy, to get those accuracy numbers up. We're building one of these single bit or SNP callers. It's called BigE, and it's written in Spark. And we've got a structural variant calling. What's interesting about this field, because kind of only God knows what your genome is exactly, because there's measurement errors in the tools, the biological tools, as well as the alignment, so nobody's confident. So we have to figure out a way, how can we do benchmarking to measure progress when we don't have we don't know what truth is. So that's an interesting challenge for us to do. We know how to do performance benchmarking, how to do that properly, but how are we going to do it when the accuracy is flexible? We'll talk about that. And we've been concentrating on the normal cells, but we're motivated to do cancer, and cancer is going to have much more variety, that the structural variants are going to be much more prevalent in this bizarro cells of cancers than they are in the normal cells, and so we're going to bring it to that. Okay. I think what I could do in our luxurious the advice was for me to do 20 slides and leave less time for questions. So I will talk about the repository piece next, which is different than this. But are there questions about the pipeline or SNAP yet? And then if I get questions, or otherwise I'll just keep blowing ahead here. Yes? You have to be a little louder. Where's the genome's data stored at? I'm going to talk about that next. Other questions? All right. Go ahead.
Do you envision scientists using your stack, writing their own software on top of your stack?
Yeah, our idea is we want scientists to use it, of course. It's open source. Like everybody, we want people to use our software. Our idea of following the software-oriented architecture, it needs to be easily extensible, so that without a lot of work, they could add things into it. So yeah, our vision would be, we'll make this available, and there's all kinds of people. This is kind of the low level. We're trying to tell you this is what the genome looks like, and there's all this pathway work that we talked about. There's lots of other levels that would be built on top of it, and we're hoping scientists would do that. It'd be great if they would use more productive tools like Spark. We're hoping that we could convince them of that. When I visited a meeting and asked what software, I was trying to figure out about the software, and they're using, sed scripts and all kinds of terrible things to a lot of Perl scripts and stuff like that. So I'm hoping they'll use more modern languages. But whatever they want to do, we'll make it easy to interface a new software. Well, you want to get more information about what we're doing? Yeah. If you go to the AMPLab website, we have a bunch of projects on the AMPLab website, like Spark and things like that. You can also find, it's either called Under the Cancer Genomics or about SNAP itself, that we have a web page for that. So if you're interested in those things, okay? All right. I'm going to get a suntan when this thing's over here. All right. So great. Maybe we'll get people together and build a better pipeline, and hopefully people will use it and extend it. But to create that vision of the future that we talked about, we need to have a repository, right? We need to put not only the genetic information, we need to have the patient's electronic medical records, and then what therapies were tried and so forth. So last week, we published a white paper on this topic, and we being the people in the AMPLab, as well as people at UC San Francisco, and we collaborating with UC Santa Cruz. David Hausler, who's one of the heroes of the Human Genome Project, is working with us on this thing. And he said it's time for us to write a white paper to kind of lay out not only the policy issues around storing all, having such a repository, but the technical issues as well, to try and explain to them that it's affordable. And so you can find that white paper yourself, and it does have an executive summary if you don't want to read the whole thing. So one question is, why 1 million? So we have machine learning experts there, statistically well-grounded people, including David Hausler, and he says, we need to get to 1 million before we have enough samples. Cancer, well, we think of it as lymphoma and breast cancer. There's really lots of subtypes. So to have enough samples that we could plausibly help individuals, that once we have the information in there and you come, there'll be examples of cases that have your subtype and treatments. He thinks we need to get to 1 million cases. So that's why we say 1 million genome project, 1 million cancer genome project. So what we have right now that David Hausler has led is he has built his own cluster in a data center to do what's called the Cancer Genome Atlas, TCGA, which has 20,000 tumors, the genetic information from 20,000 tumors, but not very little patient information. So it's been collected from all those cancer centers from all over the country, and the warehouse itself, even though it's at UC Santa Cruz, is kept at the supercomputer center at UC Santa Barbara. This is what it looks like. It's got about 5 petabytes of information there. And so what did they learn? Basically what they learned, this is really hard to run a storage system. It's basically what they learned. It's expensive, it takes special expertise. The compliance that you need, because it's medical information, can be time-consuming. The data security thing's hard. How do you do backup with petabytes is hard. So basically, and although the bigger it gets, the easier it gets to manage. But basically, they did this as a kind of responding to a national need as good citizens. It wasn't the thing that they wanted to do, but they see the reason they did it. But having that experience, that led to the white paper, what about we should do in the future? And as everybody here knows, we need to have the computation near the data. It costs a lot more to send the bits to the computers remotely than to keep them all together. So obviously cloud computing, this whole conference has been about the advantages of the cloud versus everybody having their own data center. And so we estimate in the white paper that for about $25 a genome a year, you could store it to have at the scale of 1 million. So that's, that's our estimate. But if you think about it, if it's $1,000, right, hopefully in a few years it'll get down to $1,000 to sequence it, $25 a year for it to keep around now, and of course that price will drop. Seems like this is something we should be doing soon rather than sometime way off in the future. They did learn some things about trying to put this together that might not have been obvious to people everywhere. The first is a lot of this information comes from clinical trials. So typically, the patients will agree for the researchers associated with that trial, they'll sign this consent. Researchers associated with the trial can look at the data. Well, if we want to get to 1 million genomes, which would be 1,000 trials, it'd be crazy to have every researcher have to get permission from 1,000 different boards to see this data. I mean, that's crazy. And that would make us an unattractive data set. So we should have a single national consent form for cancer patients. And it doesn't matter which hospital they go to, they sign this form so that any researchers can use it. Now, a lot of this work is being done by researchers, and as noble as researchers certainly are, maybe they would like to publish, before they put their data in, they'd like to have the right to publish papers on their data that they spend a lot of time collecting. So what they did in Hausler's, what's called the Cancer Genomics Database, the thing I told about, they give you 18 months to get your paper published, or whenever your paper is published, then other people. So everybody can see the data, they just can't write articles on it until you do, or 18 months expires, but that seems to work fine. There are people who are trying to file patents on genomes and stuff like that. That would be a problem if you have this community resource and people are trying to race to file patents. So that's not you can't do that with the Cancer Genomics Hub, and we think that's a good model to follow. You could just restrict it to the in-crowd that can work on it, but that's, that might help the in-crowd, but it can't be good for humanity to restrict it. So the policy that they have and that I would hope would happen for the 1 million cancer genome would be any researcher can access it. And then finally, they tend to be point studies. This was this cancer at a particular point of time. It'd be a lot better to have longitudinal studies to see what happens to the cancer over the years, and which is not supported right now. So how do we get there? We're talking about tens of millions of dollars, even at only $25 a genome, and I don't think they're going to decide to do that based on a white paper that Hausler and I worked on. So what we think will happen is we need to get some smaller studies to start showing some value, and then as we see value, then we could grow it, and then we could get to the 1 million genome case. So the first one we call patient push. So it turns out cancer patients own their electronic medical records, and if their DNA was sequenced, they own that as well. So an idea would be to form a national nonprofit as a repository, and they would go through patient advocacy groups to find patients and get the data from the patients themselves. So they go around the medical centers and get it that way, and we'd end up with this one big repository. And you can imagine that a lot of cancer patients would love to have their data contributed so that we could help find cures sooner, and maybe even them. The other is kind of the opposite, to go to the cancer centers and ask them to contribute, the enlightened centers who can see the advantages of putting a lot more information together so they can help to the cure. And it turns out that the top few centers, which are listed there, MD Anderson and the Memorial Sloan Kettering Cancer Center, these top few centers handle 10 or 20% of all the cancer patients in the United States. So there's a handful that dominate here. And they could get maybe more elaborate medical records. But basically what we argue in this paper is, this is so important, we don't know that there's a single right path. We should probably pursue both paths. And so we could agree on standards and so forth. Okay, so this is my last slide. So the AMP project is a big data project. What we just needed was a lot of data, and that led us into this very humanitarian thing. Cancer is this amazing, scary disease. The more I read about it, the more it frightens me. I have this sense that it's really urgent to work on this problem for selfish reasons. I need those answers out there soon. The genetic sequencing is this amazing technological success story. A factor 10,000 drop in 6 years, soon under $1,000 a genome, which as a result, we're going to have a lot of data that comes out of it. It hasn't happened yet, but there's a tsunami that's on its way. And amazingly enough, people with backgrounds like us could help people with cancer or help people not cope with the cancer. Our software skills could help build better pipelines. And we could help with this repository. So and when you think about the numbers that I'm talking about, if we could move this future 5 years forward, so it's 600,000 people in the United States die every year. If we could prevent half of them dying, so 300,000 times 5 years, 1.5 million people, that is as many people who have died, as all the soldiers in all the wars in the history of the United States, is 1.5 million people. That's like every 5 years we could make that happen sooner, we could save equal to all the war deaths. So, what an amazing thing that there's a chance, right, that we could help. And like I said, if there's a chance we can help, as moral people, aren't we obligated to try? All right, I'm ready for more questions.